Hi, this is going to be a very quick review for Exam 1 in Biology 1406 lecture. You should absolutely reference the exam review sheets that already exist in Canvas. Again, this is just a very quick verbal review. What I've got is my notes for chapters 1 through 5, and I'm just going to go quickly through each of the chapters and let you know highlights of things you absolutely should know for the exam. So again, this is not meant to be comprehensive, just to give you an idea, especially those of you guys that like videos. Here you go. So, Chapter 1, you definitely need to know that evolution is biology's core theme. You should know that biologists ask questions. Know the 7 characteristics of life and be able to understand what each of those 7 elements are. Know the 10 levels of biological hierarchy or biological organization. Know how they fit one another, which one's bigger, which one's smaller, be able to provide examples and be able to apply them to scenarios. With the level of a cell, you should understand that that is biology's fundamental unit of structure and function. You should be able to describe the different or the 4 characteristics that all cells have in common. You should know the difference between prokaryotic and eukaryotic cells. You should know what is meant by the word gene expression or biology's central dogma, that DNA makes RNA that makes a protein. You should be able to talk about negative and positive feedback mechanisms and understand which is which. Concept 1.2, know taxonomy. Did King Philip come over from Germany stoned? What are those different layers? Within the 3 domains of life, if we do similar to what we've had on quizzes and in class where I describe an organism to you, you should be able to classify it into the appropriate domain, and then if it's a eukaryote, divide it up into the appropriate kingdom or whether or not it's a protist. You should understand Darwin's main 2 points and the observations that he made. You should understand that in evolution, natural selection is the process that is doing the evolution. Concept 1.3, you should understand what science is. You should be able to differentiate between quantitative and qualitative data. Make sure that you understand what a hypothesis is, be able to go through the scientific method. Make sure that given an experiment, you can identify the independent and dependent variable, know where those would appear on graphs. Be able to differentiate between hypotheses and theories. Concept 1.4, be able to differentiate between science and technology. Science is discovery, technology is inventions. Again, you can see this is going to go pretty fast. Chapter 2. Chapter 2, know what an element is, know what matter is. Be able to describe what a compound is. Know that we require about 20 to 25% of the elements for life. The big 4 are carbon, hydrogen, oxygen, and nitrogen. For those 4, make sure you know their valence, make sure you know their bonding capacity, make sure that you know their chemical symbols. Most of the remaining of us is elements such as calcium, phosphorus. Know what trace elements are, those that we require in small amounts in order to survive. Concept 2.2. Know the charge of a proton, neutron, and electron. Know where those are found as we look at an atom. Know the electrons are not going around in perfect circles, but instead are traveling around in 3-dimensional spaces called orbitals. If I provide you with the atomic number and the mass number of an atom, you should be able to determine the protons, neutrons, and electron count of that atom. You should also feel comfortable drawing that electron structure as we've been practicing in class. The number of protons is important because it determines what type of atom we're looking at. The number of neutrons is important because this can change an atom into various isotopes. Electrons are the exciting part, however, because electrons determine the binding capacity or how an atom is going to behave with other atoms. With electrons, you should know how to fill them in. 2 electrons go in that first energy shell, then 8, then 8. With electrons, be able to differentiate between valence and then this other thing called the valence electrons. Know the valence for carbon, hydrogen, oxygen, nitrogen. The valence of hydrogen is 1, the valence of oxygen is 2, nitrogen is 3, and carbon is 4. Know again that electrons are going around in 3-dimensional spaces called orbitals. Concept 2.3, we talked about 4 different bonds or interactions. The first type of bond was a covalent bond. You should know that this is a strong bond where we're sharing electrons. Covalent bonds are what we call molecules. With covalent bonds, you should be able to differentiate between polar and non-polar covalent bonds. A non-polar covalent bond, the electrons are shared equally, so there is no difference, no charge. In a polar covalent bond, something like water, the electrons are shared unequally because something is electronegative, creating a charge or a polarity to the molecule. Please understand how to draw a water molecule, understanding where the charges are on that water molecule and where that polar covalent bond is. With ionic bonds, it's a weaker bond. Know everything about sodium chloride, which one's the cation, which one's the anion, which one's losing or gaining the electron. Another type of weak chemical bond that we talked about was the hydrogen bond. Make sure that you understand that hydrogen bonds form between molecules. If given two water molecules, be able to appropriately diagram where that hydrogen bond would form. And then our last bond that we talked about was the van der Waals interactions. And remember with that one that it has an asymmetrical electron distribution. Know that molecular shape is important to function. Triangles fit into triangle slots, circles fit into circle slots. And then the last concept, 2.4, we talked about chemical reactions. Note the starting molecules are called reactants, the end products are called products. Know how to look at an equation and tell if it is balanced or not. And know that chemical equilibrium is when the forward and reverse rates of the reaction are equal. Chapter 3. Again, you should be comfortable with a water molecule, knowing where the charges are, knowing how it will form hydrogen bonds with other water molecules. Know the 4 emergent properties of water, and then understand why each is important to biology. So the 4 emergent properties are: number 1, it's cohesive; number 2, they have the ability to moderate temperature; 3rd, it expands upon freezing; and 4th, it's a versatile solvent. So with cohesion, make sure you differentiate between cohesion, adhesion, and surface tension. With number 2, the moderation of temperature by water, you should know the difference between heat and temperature. You should know how heat moves. Make sure you understand the Celsius benchmarks, 0, 25, 37, and 100 degrees Celsius. Know what water's high specific heat is traced back to and why this is important for biology. Know what evaporative cooling is. Number 3, floating of ice on liquid water. Understand that ice is less dense than liquid water. Know that water reaches its greatest density at 4 degrees Celsius. Number 4, water is a versatile solvent. Know the difference between solutions, solvent, and solutes. Know what an aqueous solution is. Know what hydration shells are and how water is easily able to dissolve ionic compounds. Differentiate between hydrophobic and hydrophilic substances. Be able to tell me how to make molar solutions of various molecules. And we've been practicing those a lot in class, but you should know how to make molar solutions. Know what Avogadro's number is, that 1 mole always equals 6.02 times 10 to the 23rd molecules. Concept 3.3, know about how water dissociates. Know that an acid increases the hydrogen ion concentration of a solution. A base is going to decrease the hydrogen ion concentration of a solution. With the pH scale, know the relationship that the concentration of the hydrogens to the concentration of the hydroxides always equals 10 to the negative 14th. Also know that pH is the inverse log of the hydrogen ion concentration, and be able to use those formulas similar to the ways that we've been discussing in class and that you've seen on the quizzes. Know what a buffer is. It's able to maintain a set pH. It's usually a weak acid-base pair. It's not neutralizing things, it's trying to maintain some set pH. Know what ocean acidification is, that as more CO2 goes into the water, that acidifies, and how that impacts the environment. Chapter 4, know what organic chemistry is. Know who Stanley Miller was and what his experiment showed. Know that carbon can form up to 4 covalent bonds with other atoms. Know that carbon has a big diversity. Know what a hydrocarbon is. It's something made out of hydrogens and carbons only. Isomers are molecules that have the same molecular formula but different structures and properties. The 3 different types of isomers are structural isomers, which vary in some sort of branching form, cis-trans isomers, which vary around a double bond, and enantiomers, which are mirror images of one another. Know the chemical functional groups. All of the functional groups are going to be important because they are hydrophilic, meaning that they're going to increase the solubility of items in water. Hydrocarbons on their own are mainly made out of non-polar, so no charged covalent bonds. So most hydrocarbons don't like to dissolve in water. So if you want to get a hydrocarbon to dissolve in water, you're going to need to add some functional groups to it. These are really important, as seen in the example that we looked at as estrogen and testosterone. For the 7 functional groups, make sure you know their names, how to draw them, what is important about that functional group, and then how to name compounds that contain that functional group. Chapter 5, structure and function. Know the 4 broad classes of macromolecules that we studied, their monomers, and the bond names of each. In exercise 5.1, we set up some basic rules, differentiate between polymers and monomers, and know the difference between dehydration and hydrolysis reaction. In concept 5.2, we went through a variety of carbohydrates. You should know the structure and function of the following: the structure and function of monosaccharides, the structure and function of disaccharides. We talked about 3 disaccharides specifically: lactose, maltose, and sucrose. For the polysaccharides, you need to know the structure and function of starch, the structure and function of glycogen, the structure and function of cellulose, and the structure and function of chitin. We then switched to the lipids. Remember that lipids do not have monomers. They don't fit that monomer-polymer discussion. However, we talked about 3 different types. You should know the structure and function of fats. Make sure you can differentiate between saturated and unsaturated fats as well as trans fats. Then make sure you know the structure and function of phospholipids and the structure and function of steroids. Concept 5.4 with proteins. Functionally, their function is anything. Their function is very diverse. Structurally, make sure you know the difference between a protein and a polypeptide, be able to draw an amino acid, and be able to talk about the difference between primary, secondary, tertiary, and quaternary structure. Primary structure is held together by hydrogen bonds. Secondary structure is going to occur because of interactions that happen between the backbone units, and then tertiary structure is going to happen because of interactions that happen between R groups. Some molecules are complete once they have tertiary structure. However, some polypeptides require multiple subunits to come together before they are functional proteins, and that's called quaternary structure. Make sure that you understand how sickle cell disease was an example of how important protein structure is when it comes to human health. Know what a chaperonin molecule is. You know what? I think I pulled that out of the lecture, so don't freak out about chaperonins. All right. Concept 5.5 is nucleic acids. Know the monomer of a nucleic acid. Know their functions. So the monomer of a nucleic acid is this thing called a nucleotide. Know the 2 functions of DNA and the 2 functions of RNA. With the structure of nucleic acids, they're made out of nucleotides. Be able to differentiate between pyrimidines and purines, and know whether we use deoxyribose or ribose to make up DNA or RNA. Know that DNA is a double helix made up of 2 strands that run in an anti-parallel arrangement, whereas RNA is single-stranded. Know how base pairing works within DNA and also how base pairing works from DNA to RNA. In concept 5.6, you should differentiate between genomics and proteomics. Again, that's a really fast version of a review. Please use the complete review sheet that exists in Canvas, but that might give you a place to get started as you start preparing for exam 1. Thanks.